So let me introduce Laura real briefly. I'm very excited to welcome Laura. She's jointly appointed between Stanford Data Science and the Tsai Institute and the Department of Psychology. As I mentioned several times, her work is focused on understanding the neural representation and operations that give rise to speech comprehension in the human brain. So this is very exciting. And she's going to use data and machine learning to achieve these goals and take advantage of recording techniques that operate at many different spatial scales. We cannot be more pleased to have you, so I'm going to give you the floor and welcome you officially to Stanford Data Science.
Thank you. Thank you so much. Great. Thank you so much. Thank you for the introduction. It's so great to be here. It's really an honor to get the chance to share my research with you all. And I'm really excited to talk to you about some of the work that I've done so far and some of the future directions that I'm going with my new lab here at Stanford. And as Emmanuel said, I'm an assistant professor in the psychology department. Very honored to be a faculty fellow here at Stanford Data Science, and I have a faculty scholar appointment at Tsai Neurosciences Institute as well. And the goal of my lab is to understand how the human brain achieves language. So what is the algorithm that allows humans to verbally communicate? So what's going on in your brain right now that allows you to understand, hopefully, the words that I'm saying to you? And how can we use advances in artificial language systems to help us to unveil that process? And then how can we, in turn, use our understanding of the brain's capacity for language to actually improve those artificial systems? So my research lies at the intersection between a number of different disciplines. Neuroscience, because I want to understand how the human brain achieves language processing. Linguistics and cognitive science, because this provides rich theories for how the human brain achieves this. And data science and artificial intelligence as performance-driven methods for building algorithmic theories of how the brain achieves language processing, as well as a powerful data analysis methods for being able to handle the rich multivariate neural time series that we can record from the human brain. Okay. And I want to make it very clear that this, right now, is a super exciting time for language neuroscience, for a number of reasons, but for two big reasons in particular that I wanted to highlight. So the first is that we're currently at a paradigm shift in terms of the type of neural recording technologies that are available for human neuroscience. So on the one hand, there's been really amazing improvements in our ability to record neural signals non-invasively. So the optically pumped magnetometer system, for example, or OPM, is a way of being able to record neural activity with millisecond resolution and millimeter resolution from putting sensors placed outside of the individual's skull. And on the invasive neural recording side, there have been really amazing advances as well in nanotechnology, which give us the ability to record single neurons at large scale, so hundreds of neurons at the same time, across the cortical depth using neuropixels probes. And I'm going to actually present some of the data that we can get from this neural recording technique today. Okay. So this is one big reason why language neuroscience is at a really important stage. And the other is that for the first time in history, there exists a system other than the human brain that is able to operate with language. So here, this is the response I got from ChatGPT when I gave it a prompt that I think it had never heard before. So please describe in the style of Shakespeare what language neuroscience is like in a modern AI era. And it did pretty well. And the point of this is just to say that this is the first time that we're able to actually work with artificial systems that can respond to text prompts in appropriate ways and provide coherent text in response. And this is exciting for language neuroscience because these serve as rich hypothesis generators for the type of processes that maybe the human brain is also using to process language, as well as a powerful analytical tool to summarize very complex properties of language, like the meanings of words and sentences, and actually condense that down into a numerical representation that then we can then use to test in our neural data. Okay, so with these two amazing advances in hand, this will allow us to use these advances in neuroscience not only to advance our understanding of the neural encoding of language, but also to use that insight to in turn improve artificial systems. And we can then also use these incredible advances in artificial systems to better understand the neuroscience of language. So today I'm going to present some of my work today that focuses in on the case of speech comprehension in the human brain. So in speech comprehension, the problem that the brain needs to solve is to take the auditory signal of speech and from that derive a conceptual understanding of what it is that's being said. And in the case of speech production, we're essentially having to do this process in reverse. We have a concept, an idea that we want to convey, and we need to translate that idea into a coordinated sequence of speech sounds. So speech processing is a process of translating either sound into meaning or meaning into sound. And I've been working on understanding the processing system that allows this translation to happen. And in order to make sure that I'm making measurable progress towards this goal, I break this algorithm down into three parts. So the first is, what representations does the human brain use in order to achieve this mapping from sound to meaning? And some examples of representations might be an acoustic representation of the input that we derive from the ear. Or it might be something like the syllables or the meanings of the words that are being said. We also need to understand the operations. So operations might be filtering of the acoustic signal within a certain frequency range, or applying a nonlinear categorization. And then finally, we need to know essentially the order with which these different representations get generated and the order with which these different operations are applied. And so this tells us how the information is actually passing through the system. Do we have feed-forward connections, feedback connections, recurrent connections, skip connections, etc.? Okay. And how are we going to be able to understand these different components of the processing algorithm? Well, if we're working with artificial systems, we actually have access to every single unit in that system. But for the case of human neuroscience, we need to record neural activity. And as Emmanuel nicely alluded to, my lab works with neural recording techniques at different spatial scales. So on the one hand, we have the ability to record activity across the whole brain using non-invasive methodologies. Going one level further, we can also record neural activity directly from either the cortical surface or inside of the brain by working with individuals who are undergoing clinical treatment. And most recently, I've also worked with neural recording methodologies that allowed us to record from individual neurons' spikes. And there are two techniques that I want to highlight here. One is NeuroPixels probes, which allow us to record across the cortical depth. And the other are UTAR arrays, which actually have lateral XY coordinate system. Okay, and I think it's really important to operate with neural recording methodologies at these different spatial scales so that we're certain that the theories that we engineer are able to satisfy multiple spatial constraints. So we want to come up with a theory that not only satisfies the data at the whole brain scale, but also satisfies the data all the way down to single neurons within cortical columns. Okay, so in the first part of the talk, I'm going to show you some results where we're using whole brain non-invasive recordings. And the goal here was to focus in on this question of neural representation. Okay, so as I said, during speech comprehension, the human brain needs to map sound into meaning. And this mapping is largely arbitrary. So, for example, I could convey to you the same meaning in, say, 3 different languages. And in each of those 3 languages, the sounds that I use and how they're coordinated are going to be quite different, even though on the meaning side, they're very similar. And so in order to bridge this arbitrary gap, the brain needs to transform the acoustic signal into a hierarchy of representations that with each transformational step, the information becomes closer and closer to the meaning of what's being said, and further away from the actual sensory realization of what was said. So in this first study, we'll talk about, first of all, what are the representations that the brain generates in order to map from sound to meaning? And second of all, in what order are these different representations generated when people are listening to continuous natural speech? So I asked 21 native English participants to listen to 2 hours of short stories while I recorded their neural activity with whole brain magnetoencephalography. And every 4 minutes or so, they had to answer a comprehension question about what they were hearing, just to make sure that they were engaged and understanding the stories. And I actually have since publicly released this data set, because this is a very valuable set of data that one could use to address a number of different questions. So if you have need for such a data set, then I recommend you go look at that. Okay, so the data that we get from an MEG system essentially looks like this. So the person has 208 magnetometers placed all around their head. And each magnetometer is taking a measurement every millisecond. So that's the time axis here. So we're recording their neural activity continuously over time as they're listening to these stories. And I annotated in those stories when each of the important moments in the stories happened. So in this case, when each word in the story occurred. And that's what allows me to align this continuous neural data stream to the moments when each word in the stories occurred. Okay. And in order to get at this question of what representations are generated, I annotated those stories, not just for when each word occurred, but what the various properties of those words actually were. And I annotated them for 50 features in total, which span from the lowest properties of speech, so essentially sound, all the way up to meaning. And I group those features into 6 feature families. So we have properties of each individual speech sound, which we call phonetic features. Sublexical properties, like how many syllables are contained in this word and how many morphemes are in this word. It's kind of a measure of lexical complexity. Word class, otherwise called part of speech, like is this a determiner, an adjective, a noun, a verb, etc. And then properties related to the actual sentence structure itself, like how many branches of the tree can we create at a particular word and how many can we close? And how deep into the tree structure does a particular word occur. So these are measures of sentence complexity, word by word. And as you can tell, these are all features that are very interpretable, and they have been derived based off of cognitive science and linguistic theory. And at the highest level, we have proxies for semantic representations using glove word embeddings, which are an unsupervised method of deriving word vector representations, which map onto different meanings of the words that people are hearing. So the question is going to be, when I fit a ridge regression model, where the input to this model is the strength of neural activity across those 208 sensors, how well can I actually reconstruct the properties of language that people are hearing? And in what order can I reconstruct those properties? So just to make the order question a little bit more concrete, I'll show you the two extremes of the hypothesis space here. So one hypothesis says that language is compositional. So we have phonemes, you combine those to make syllables, you combine those to make words, you combine those to make phrases, etc. And so because language is compositional, language processing is going to be compositional as well. If that's the case, the result of my ridge regression should say that I can decode the lower level properties of speech, like the phonetics, earlier than I can decode the higher level properties of speech, like those semantic vectors. The other extreme of this hypothesis space says that, well, actually, because people are listening to continuous speech, they can actually anticipate or predict the meanings of the words that they're going to hear before they actually hear them. And so if that's the case, we'll actually be able to read out from the neural activity the higher order properties of speech, like the meanings of the words, before we can read out what those words actually were in terms of the individual speech sounds that were said. Okay. So let's take a look at what the data support. I'm going to show you the results of decoding each of these 6 feature families on the right-hand side. And we have time on the x-axis relative to the word boundaries in the stories. And I'll first show you the results of decoding the properties of the speech sounds in the stories. So I'm using a stack plot here because there's multiple features in that feature family. And this solid bar shows you the result of a statistical test that tells you when we can decode this property of speech better than chance. So we can decode the phonetic features from about 0 to 200 milliseconds, sublexical properties from about -100 to 500 milliseconds, word class from about -200 to 200 milliseconds, syntactic operations from -200 to about a second, and the syntactic state and the semantic vectors were decodable from about half a second before the word ended until a second and a half afterwards. Okay, so what this tells us, first of all, is that all of these features that have been proposed by linguistic theory and cognitive science in the past, actually all of them seem to be encoded in neural activity of these people listening to natural stories. And we also see some interesting things in terms of the dynamics with which those representations are encoded. So we see that the higher order properties of speech indeed do seem to be encoded earlier than the lower level properties. And there's also an extended period of time with which this entire hierarchy is actually all encoded in the brain at the same time. I'm not talking here about which particular parts of the brain are encoding these different features. That's a future direction that we're working on. But across the whole brain, all of these representations across the whole hierarchy are coexisting in neural activity. Okay. So if I pull back then the two hypotheses, it's clear that the hypothesis that wins out here is this reverse hierarchical approach. And thank you for the murmurs of the 90s animation. So it's very interesting that this is the hypothesis that wins out, because it's been primarily put forward for visual processing in the past and not really put forward for auditory processing or speech processing. So what this hypothesis has to say for itself in the visual domain is that the visual inputs that we receive are actually very noisy and ambiguous as to the higher order objects that we're trying to recognize. And so it's actually a very good processing strategy to use the context in which we receive those visual inputs to guide and bias how those inputs are actually interpreted. And so I think that the same thing is happening here for the case of speech processing, that we use the context in which we hear someone talk to help to disambiguate what it is that they're actually saying. Okay. And I just want to take a moment to point towards some of the future directions that currently are ongoing in the lab. So as I said, here we're using a basis set of features that are very interpretable and grounded in linguistic theory. But we can essentially only go so far with that feature set. So there's some great work going on in my lab by Mariana, who's sat right here, who has been working in the domain of explainable AI. So we want to try to understand what representations are speech and language models using to solve a task. Is there any shared representational space between those models? Can we understand what those representations are? And can we then find evidence for those same representations in neural activity? Another axis of work ongoing is to get a better understanding of the spatial encoding of this different piece of information. So here we also want to push to representations across longer timescales, so the meaning of what's being said across multiple sentences using large language models to be able to represent such a complex property, and to understand how this plays out in the spatial localization of these features. And then finally, Irmak, who's my graduate student, is working to better understand specifically the semantic axis. So there are many different ways of representing meaning using word embeddings. And we're doing a deep dive to try to understand how can we derive semantic representations in different ways, what do these representations bias, and how does that get encoded in neural activity. Okay, great. So now I'm going to shift gears a little bit to talk to you about neural data at the other spatial extreme of this continuum. So here we're looking at data from NeuroPixels probes, which, as I said, give us the ability to record spiking of individual neurons in human cortex. So with non-invasive methodologies like magnetoencephalography, we have the ability, as I said, to record from the whole brain. However, if we're using an invasive method like NeuroPixels, we need to decide where it is that we're specifically going to target. And so in this work, we decided to target superior temporal gyrus because it has been implicated a lot in its role for speech processing. So neural populations in this area encode phonetic properties of speech that we already discussed, as well as other types of speech properties like fluctuations in the loudness of my voice or the pitch of my voice. And when you record neural activity in this area from the cortical surface, you get a very nice tiling whereby different neural populations encode different types of speech features. But what we really didn't know anything about is how the neurons underneath any one of these XY coordinates actually give rise to the sensitivity that we record from the surface. So one hypothesis is that, well, all of the neurons in a cortical column are all going to be tuned to the same thing. And that's the same that we're going to observe the sensitivity from the cortical surface. The alternative says that actually neurons in a cortical column are going to be tuned to various types of speech features. So maybe there will be a dominant feature that explains the sensitivity we record from the surface, but that's not all that will be in the column. There'll be other types of speech features encoded there as well. So that can be summarized into two questions. The first is, what speech features are encoded by single neurons in this brain area, and how is that encoding organized as a function of cortical depth. But first, I want to tell you a bit more about these neuropixels probes and how they allow us to record this neural activity. So these probes have been really groundbreaking for work with animal models. They were first introduced in 2017 and have led to a huge mass of amazing discoveries. So you can see that the probe itself is extremely thin. It's only 70 microns wide, and it's 1 centimeter long. And this gives you the ability to record from hundreds of neurons at the same time, whereas previous methods, you'd be recording from a couple of dozen. And in my prior work, I helped to translate the use of neuropixels for use in humans. And this is specifically done in the context where someone needs to undergo brain surgery to treat epilepsy or a tumor. And the probe is inserted into tissue which is about to be resected as part of their clinical care. So there were 5 individuals who volunteered for this study. And as I said, they were all undergoing brain surgery in order to treat epilepsy or a tumor. And we insert the probe into tissue that's about to be resected, and we insert the probe for about 10 minutes. So during that 10-minute period, they listen to 200 sentences, and 100 of those sentences we play just once, and the other 100 sentences are 10 sentences that we repeat 10 times. And I'm saying to you this now because it will be relevant in a moment. And all of those sentences get randomly shuffled. We don't play the same sentence one after the other. And here is a schematic of the probe insertion for the first participant. So we insert the probe so that there is some contacts in white matter and some contacts in air so that we're certain we're recording from the full cortical depth. And because this tissue is about to be resected as part of their clinical care, it actually allows us to perform histology Nissl staining so that we can get an estimate of the cortical layer boundaries. Okay. So now I want to show you some of the data from this recording technique. So one of the sentences that the participants listened to was, it was nobody's fault. Here is the phonemic transcription, the waveform, and the spectrogram of that sentence. And here I'm showing you, this sentence was repeated 10 times, as I mentioned, and here are 6 repetitions. So each of these red lines corresponds to one spike from this neuron, and the x-axis matches the acoustic axis above. So I'm going to play you now 4 repetitions of this sentence with the spikes of this one neuron overlaid on top. Let's see if this works. Okay, that doesn't quite work.
It was nobody's fault.
Okay, let me just do that quickly one more time because that didn't quite work.
It was nobody's fault. It was nobody's fault. It was nobody's fault. It was nobody's fault.
Okay, so what I want you to appreciate here is that every time this sentence is played, this neuron fires at the same time for the same duration. And we can look at the responses of this same neuron now for a different sentence.
Junior, what on earth's the matter with you? Junior, what on earth's the matter with you? Junior, what on earth's the matter with you?
Okay, so this same neuron is now firing at a different moment in time as compared to the other sentence. But again, it is firing very consistently at the same moments in this sentence, suggesting that it's actually tuned to something in the input. And this consistency of firing is not just something we observe for one neuron. Here I'm showing a few examples, a representative of the hundreds of neurons we were able to record. So here, each column is a different sentence, and each row is a different neuron. And we were just listening to this neuron, for example. But what you can see is that Different neurons fire at different times to the same sentence, suggesting that they're responding to different properties of those sentences. And one neuron will fire at different times to different sentences, also suggesting that the timing of its response depends on whether the feature that it's tuned to actually occurs in that sentence and when it occurs. Okay. And we can statistically test what these different properties, what these neurons are tuned to in the speech. So we fit a temporal receptive field encoding model using all of the features that I'm showing here. So we have what are the individual speech sounds that are occurring in the sentences, what's the fluctuation of the loudness of that person's voice, the pitch of their voice, whether they stressed certain words in the sentence, and the probability of each word occurring in that sentence, and the probability of each speech sound occurring in the sentence. And this allows us to establish what these neurons are tuned to and how that tuning is organized across the cortical depth. So here's a summary figure showing you the result of that analysis, aggregating the data across participants. And you have depth of the probe on the y-axis, and the color of each of these dots, each dot is a different neuron, and the color of the dots tells you what feature best explains the activity of that neuron. And what we see is that some features are very dominantly represented across the population. So there's a lot of encoding of pitch and acoustic phonetics. And even if you look at the responses in one participant, so in just one column, you actually see the encoding of both of these features together. It's not that in some participants there's just pitch, and in other participants there's just acoustic phonetics. And what's important relative to the organization question is that these features are not randomly scattered around. We find that the neurons which encode pitch tend to localize more superficial than those which encode acoustic phonetic features, for example. So there seems to be depth-wise organization. Okay, so to summarize the main take-homes of this work, by having access to this depth axis, we find that in comparison to what's been found in animal models with, say, monkeys in primary auditory cortex, where all of the neurons in a column are tuned specifically to the same thing, here we observe a lot of heterogeneity in the neurons tuning. And we see that this tuning is not randomly scattered, but there's organization across the cortical depth. And we also see that the heterogeneity of tuning actually operates at different time scales. So there's simultaneous encoding of fast features, like each individual speech sound, which unfold very quickly, and slower features, like the pitch of the person's voice. And all of this comes with the larger take-home message that by being able to record neural activity across the cortical depth, we have the ability to be able to actually understand the circuitry of the neural processes that are ongoing in each of these cortical columns, which was essentially just inaccessible before. And this project is also really, it represents this paradigm shift, as I said, of the types of data that we're going to be able to record in human neuroscience. And I think it gives a lot of exciting promise for the future. So for example, now we would have the ability to compare spiking neural networks to a biological ground truth with the vision of building data and energy efficient analogs of the leading AI systems of today. And also together with a bioengineering graduate student who I'm co-mentoring with Jamie Henderson, this is Yoon, we're exploring the spiking activity in higher order brain areas, such as the inferior frontal gyrus, which are more likely to implement the symbolic properties of language rather than the more sensory acoustic properties of speech that I've talked about today. Okay, so to sum up, what did we learn about this processing algorithm of language? I think that one of the big themes across a lot of my work is that there's a lot of parallel processing ongoing in the encoding of representations. And in the first study, we saw that there's a lot of parallel encoding over time across the whole brain. And when you zoom in, even down to the level of one cortical column, there's parallel encoding in space, that multiple features are co-localized. And by working with neural data across these different spatial scales, it provides a complementary multi-lens view into how these processes are implemented. So the results that we got using whole brain MEG, for example, looking at this rich hierarchy of representation, is not something that we would have been able to uncover with NeuroPixels probes, for example. So we need to tackle these questions using multiple complementary lenses. And the results that I show today, particularly the work in the first study, simply just wouldn't be attainable with classic encoding methods. We wouldn't be able to immerse people in a naturalistic listening scenario of just listening to audiobooks. And we need methods that are sensitive enough to be able to extract these very subtle representations of higher order language properties. And I've already given you a flavor of some of the ways in which we're planning on incorporating speech and language models in order to even further understand the representations that the brain is using. Okay, so last thing that I wanted to say is that a member of my team, Jill Crees, is giving a poster in the first session today. And she's talking about the work that she's done developing time-resolved machine learning methods to understand how the processes I discussed today actually can break down in cases of people with language disorders such as aphasia. So I encourage you to go talk to Jill later. And I'm not sure if there are any rising seniors amidst us today, but if the work that I presented was interesting to you, I'm teaching a capstone course next winter, and I encourage you to talk to me about it if you want to learn more. Great, so with that, I'd like to thank my collaborators in neurology and neurosurgery here at Stanford, without whom this work wouldn't be possible, and thank you all so much for your attention. I'm happy to take questions. I can hear you.
Microphones at the back, if you wouldn't mind.
Better be a good one.
Hi. Thank you for the talk. Super fascinating. I had a question about the second study of the encoding of language in single neurons. You showed us, I think, a graph of a bunch of example neurons. They seemed really diverse in when they were encoding particular parts of the sentence. I'm just curious about, we know a lot about redundancy encoding in neurons. So I was wondering, is this just a representation of the diversity, and how much redundancy was in this population of neurons that you saw? And I guess when we compare to, say, there were a lot of neurons that encoded a particular semantic feature or whatever, or vocal feature, what explains the diversity in neurons that are best explained by a particular feature, but then have different neural encoding or in time and space? If that makes sense.
Okay, thank you. That's a great question. Here, I am on purpose showing neurons that are tuned to different properties of speech, but you're right, there is a lot of redundancy. And what's interesting is that we actually observe neurons that are tuned to the same feature, but with different lags. Which is very interesting. I'm happy to talk about why I think that is. And what I maybe should also say is that within the acoustic phonetic features, you could have two neurons next to each other that are attuned both to speech sounds, but to different speech sounds. So you could have one neuron that likes M and N sounds and another right next door that likes F and S sounds, for example. So yeah, there is redundancy, but there's also diversity.
Hey, yeah, thank you. That was a great talk. Really enjoyed it. Really liked how you had very clear scientific questions grounded in theory that you use data science to answer. So question is on the first project.
Yeah.
You talked about kind of different levels of features, right, all the way from phonetic, all the way up to kind of semantic word vectors. And you talked about kind of future work. You know, it's clear like a next step is to use contextualized word representations.
Yep.
And I was just curious, you know, as you were thinking about doing that, what are the qualities of a representation that you're looking for? You know, you have many options of, you know, encoders. You can take a bird encoder, a llama, or whatever, and all of these have really been built for kind of applied use cases. But as you think about applying it in the scientific context, what are the qualities of that representation that you're looking for?
Yeah, thank you. So that's a hard question to answer because we don't really know what it is that we're looking for.
Yeah.
So one of the ways that we're trying to tackle that question, again with Mariana, is to say, okay, what representations seem to be present in all of these models? And can we focus in on those,
Yeah.
try to understand what those representations are, and then move that over to the neural space? So we're essentially taking a very data-driven approach to look for commonalities across models that were trained in very different ways, have very different architectures, but all seem to converge on a shared set of representations, and then take those representations forward.
Okay, very cool. Thank you.
Thank you. Great, thank you so much.